Water isn't just something we drink. It's the molecule that makes life possible. Discover the fascinating science behind water in biochemistry. Welcome to Biochemistry Basics, Chapter 2. Don't forget to like, subscribe, and let's explore water, pH, osmosis, and molecular interactions together.
Watch a protein attempt to fold in a vacuum. It writhes aimlessly. But introduce water, and the chain instantly snaps into a precise three-dimensional structure. The physical architecture and chemical homeostasis of all living systems are strictly constrained by the geometry of a single molecule. Water acts as a mandatory physical scaffold. The rules of this scaffold begin with shape. A water molecule forms a rough tetrahedron. Oxygen's outer electron orbitals dictate the geometry, pushing the two hydrogen atoms into a specific corner-to-corner arrangement with a precise bond angle of 104.5 degrees. Because the oxygen nucleus is highly electronegative, it pulls the shared electrons away from the hydrogens. This unequal sharing creates a permanent electric dipole, a partial negative charge on the oxygen and partial positive charges on the hydrogens. These opposing charges pull neighboring water molecules together via electrostatic attraction, forming hydrogen bonds. But these bonds are ephemeral. They break and reform constantly, with the lifetime of each connection lasting a mere 1 to 20 picoseconds. The result is a continuous, flickering grid of dipoles. This creates an inescapable thermodynamic and chemical environment that every biological macromolecule must negotiate to function. A common professional misconception suggests that oil and water separate because hydrophobic molecules possess an intrinsic attraction to one another. The math tells a different story. This is a thermodynamic ledger showing the Gibbs free energy equation. For any process to happen spontaneously, the total free energy change, delta G, must be negative. The driving force here is the change in entropy, delta S, a measure of randomness. When a non-polar, hydrophobic solute enters water, it cannot form hydrogen bonds. To compensate, the surrounding water molecules are forced to form highly ordered, rigid cages around the intruder. This rigid ordering of water molecules drastically decreases the system's entropy. The delta S drops. According to our ledger, this pushes delta G into a positive, energetically unfavorable state. The system is highly unstable. The physical solution is aggregation. The nonpolar molecules cluster together, presenting the smallest possible exposed surface area to the solvent. By sequestering themselves in the center of a micelle, they free the trapped water molecules back into chaotic motion. This massive release of ordered water causes a sharp spike in entropy. Delta S increases, pushing delta G negative and permanently stabilizing the system. This exact thermodynamic principle dictates protein folding. A polypeptide chain twists itself into a specific three-dimensional shape solely to bury its hydrophobic amino acids in the interior, maximizing the entropy of the surrounding solvent. The hydrophobic effect is an illusion of attraction. It is actually the aqueous matrix aggressively minimizing local order to maximize its own thermodynamic entropy. Water's physical influence extends beyond shaping single molecules. Solutes physically displace water, lowering its effective concentration and creating powerful gradients. This vector graphic illustrates osmotic pressure. Water moves across a semi-permeable membrane to equalize its concentration, generating a mechanical force modeled by the Van 't Hoff equation. The pressure is strictly proportional to the number of dissolved solute particles. In a hypotonic environment, where the surroundings have a lower osmolarity than the cell interior, this pressure drives water inward. Left unchecked, it will distend the plasma membrane until the cell physically bursts. Plant cells actively weaponize this physical threat. They evolved rigid cell walls to resist osmotic lysis, converting the inward flow of water into turgor pressure, the mechanical force that keeps the plant stiff and upright. The Venus flytrap manipulates this turgor pressure to perform rapid mechanical work. When an insect touches the mechanically sensitive hairs, the leaf cells suddenly release a massive concentration of potassium ions. Water immediately follows the ions by osmosis, causing a total collapse of turgor pressure that snaps the trap shut in under half a second. Water's colligative properties are kinetic engines driving biological mechanical movement. Structurally, water shapes biology. Chemically, it actively participates in it. Water molecules exhibit a slight, reversible tendency to ionize into hydrogen ions and hydroxide ions. At 25 degrees Celsius, the equilibrium constant of this reaction yields an exact ion product of 1.0 times 10 to the negative 14th molar squared. This absolute mathematical constant forms the physical basis of the entire pH scale. Free hydrogen ions, or bare protons, do not exist in solution. They immediately bond with water to form hydronium ions. Yet their movement through water is anomalously fast compared to larger ions like sodium or potassium. They achieve this velocity through a mechanism called proton hopping. Individual protons do not diffuse over long distances. Instead, a proton enters one end of a hydrogen-bonded chain, causing a rapid sequence of bond swaps that instantly ejects a completely different proton at the far end. Biology utilizes this phenomenon. Photosynthetic membrane proteins, like cytochrome F, contain a dedicated channel of exactly 5 tightly bound water molecules. The protein uses this chain to rapidly pump protons across the membrane via hopping. The hydrogen-bonded water matrix acts as a high-speed conductive grid, ensuring cellular acid-base reactions can occur at extreme velocities. Human blood plasma normally maintains a pH near 7.4. During severe metabolic stress, like vigorous exercise, muscles flood the bloodstream with lactic acid. Without intervention, this would drop the blood pH to lethal levels. This chart shows a titration curve for a weak acid. Notice the flat central zone. A mixture of a weak acid and its conjugate base creates a buffer, a system capable of absorbing added protons or hydroxide ions with minimal change to the overall pH, as modeled by the Henderson-Hasselbalch equation. Buffering power reaches absolute maximum when the concentration of the proton donor exactly equals the proton acceptor. Here, the ratio is 1. The logarithm of 1 is 0, and the pH of the solution exactly equals the acid's pKa. Human survival depends heavily on the bicarbonate buffer system, a mixture of carbonic acid and its conjugate base, bicarbonate. This system map illustrates the blood-lung interface. Unlike a closed test tube, human buffering is an open, dynamic equilibrium. When an acid load spikes in the blood, the excess hydrogen ions react instantly with the bicarbonate reserve, forming a large pool of carbonic acid in the capillaries. This excess carbonic acid quickly dissociates into water and dissolved carbon dioxide. The dissolved carbon dioxide then equilibrates into the gas phase of the lungs and is exhaled. The acid load is permanently removed from the body. By adjusting your respiratory rate, you are physically shifting the equilibrium constant of your blood. Biological survival requires continuous negotiation with the ionization of water. The immense complexity of living organisms is governed by the strict rules of the simplest molecule they contain. On the left, we see water's physical supremacy. The entropy of the aqueous matrix forces macromolecules into their functional three-dimensional structures. On the right, its chemical supremacy. The rapid proton grid allows the Henderson-Hasselbalch equation to drive real-time physiological buffering. Water acts as a chemical reactant, a mechanical engine, and a thermodynamic architect. Whether analyzing the mechanical kinetic trap of a plant or the exact folding topology of a human enzyme, the limits of biology are strictly bounded by the physics of the water dipole.
Congratulations, you've completed Chapter 2. Subscribe now and join thousands of future scientists. Chapter 3 is coming next, and you won't want to miss it.